Good afternoon, everybody. I just noticed that this is a weather just as hot as in California. So temperature-wise, we are 20 Celsius, just about 20, I think. I'd like to talk about very recent discoveries as far as the role of fumarate hydratase in mitochondrial metabolism, especially when it comes to cancer. And this is a fairly new topic which is entering the medical literature. We have been publishing several papers in collaboration with the National Cancer Institute, with the United States and the University of Arizona, which I'm going to detail in my talk. And just to start, I learned how to use this very quick, hope so. So when Dr. Szent-Györgyi got the Nobel Prize in 1937, one of the reasons why he was given the Nobel Prize was the catalysis of fumaric acid. Now, we know more about vitamin C, and we know more about the biological combustion or oxidation process, but fumaric acid is re-emerging as a very important enzyme to regulate metabolism, and when it comes to DNA stability, it seems to be a key enzyme. And I'd like to talk about this particular enzyme, its function in the mitochondria and also in the cytosol, and how it controls cell proliferation in general. Now, it's very interesting that my grandparents on my mom's side, they knew him personally, and my grandma was talking about, my grandpa was at that time stationed in Szeged, and she was talking about him. And as a young kid, I was just very eager to hear about all the stories. And my grandma told me that actually his wife complained when he was working on vitamin C that he just was not, she just couldn't talk to him at those days. So I think as far as my scientific background goes, I'm really proud of the fact that I have had family members who had known Dr. Szent-Györgyi, and I can give this talk here in Szeged, where actually fumaric acid was, and fumarate hydratase was discovered, and eventually was included in the Nobel Prize. Now, when I ask what fumaric acid or fumarate hydratase do, not many people, especially molecular biologists, would pull out just right, it's not on top of their head, but it's a very simple reaction, adding a water to fumarate. And the conversion of fumarate to malic acid is what fumarate hydratase does. And it's a reversible reaction. It's a very conserved reaction in the TCA cycle, but this reaction seems to be a very key event in energy production, in maintaining metabolite concentration, intermediate concentration in the TCA cycle. And it's also very key to transfer hydrogen from water into reducing intermediates, especially NADH and NADPH, which eventually will end up in DNA and in fatty acids of mammalian cells. We are not there yet, but eventually we're going to follow and trace this fumarate through metabolism, and we'll see how it impacts on physiology and biochemistry. As you can see, the water, as it enters fumarate, will be actually used as a hydroxyl group and also as a hydrogen attached to the molecule. And many things can happen to malic acid. Malic acid is what we know is in apple, and it can be used to get recycled in the TCA cycle in the form of oxaloacetate, but it can be also shuttled from the cycle into the cytosol. And this hydrogen can be used for additional metabolic reactions, primarily for reductive synthesis. The enzyme itself, fumarate hydratase or fumarase, is a very conserved enzyme in the TCA cycle. In fact, it's in the process of adding water to fumaric acid eventually is a key event in maintaining the cycle to produce energy, ATP, and recycling hydrogen. And there is a catalytic arm which has a lysine and a histidine residue. And this is where water and fumaric acid react, and eventually this is how fumarate gets hydrated. And what's very interesting about the process itself, that it happens in the mitochondria, happens in the citric acid cycle, but it can also happen in the cytosol, because the enzyme has cytosolic and mitochondrial locations. Yet, in the mitochondria, fumarate and the malate conversion with the use of water is a key event to eventually keep the cycle turning over and keep producing energy by recycling this hydrogen from water. And what the significance of water is what is key to understand how mitochondria plays a role in maintaining reductive synthesis and NADP, DNA, and nuclear membrane stability. So just to follow the cycle for a short term, fumarate hydratase is one of the last reactions in the cycle. If you recall, citrate is the first intermediate in the cycle, which is formed from an oxaloacetate and by an acetyl coenzyme A, and eventually by citrate synthase, citrate is formed, which is a 6-carbon molecule. And as citrate turns over, what's very interesting is that the cycle uses water to actually keep the cycle spinning, as well as dropping these hydrogens to the cytochrome system, and eventually to shovel them into the intermembrane space. And this hydrogen is very unique because it comes from the hydrogen that comes from fatty acids. And eventually, when water is reassembled using a cytochrome, the cytochrome system as well as complex 4, we're going to see that this water is very different from the water that we drink. And this is a very new and very significant model that we are now using for understanding cancer better. As you can see, the cycle itself consumes 2 net molecules of water, which it enters at aconitase and it enters at fumarase. So without this water, we would not be able to recycle fumaric acid into malic acid and eventually oxaloacetate. And this is key to understand the energy production process, because this oxaloacetate is like a water donkey. It actually turns and carries this hydrogen, or the acetate unit with hydrogens, from fatty acids or other substrates into the cycle, and eventually 2 carbons are lost in the form of carbon dioxide, but the hydrogen from this substrate is transferred into NAD, and NADH will use this proton to generate ATP or energy, which we're going to mention, or I'm going to talk about, just refresh your memory about the process in more details. So the mitochondrial version or the mitochondrial fumarate hydratase, the enzyme itself, it functions, as I told you, to actually keep the citric acid cycle and to produce NADH. And as you can see, one of the fate of malic acid, which is formed from fumarate, is to produce NADH, and this hydrogen can be used to keep this proton gradient between the membranes of the mitochondria. Now, what's really another prominent use of the cycle, and this is where cancer comes very close to the topic is that when actually fumarate hydratase is involved in the production of NADPH, or the reducing equivalent, which actually takes place in the cytosol after malate shuttling, but malate is carrying metabolic water from the mitochondria. So this malate shuttling, or the malate shuttling process, is a key event in eventually producing NADPH for reductive synthesis in the cells, and it happens through the decarboxylating oxaloacetate decarboxylating reaction, which is an NADP dependent reaction. And what happens is malate gets shuttled from the cycle through the mitochondrial malate shuttle system, and while pyruvate is formed, NADPH is produced. So here you can see that the malic acid, which carries the hydroxyl group after fumarate hydratase reaction, adding water to fumaric acid and malate, and NADP reacts, and eventually carbon dioxide is released, a pyruvate is synthesized, and eventually an NADPH, a reduced NADP is released, which is used for DNA fatty acid synthesis and also for reductive carboxylation, which I'm going to talk in more details. Yet this is a very key role of transferring this metabolic water through fumarate hydratase to malate, and malate transferring this hydrogen to NADPH, which is a major reducing source of cells or in cells. And there is a cytoplasmic fumarate hydratase, as I told you, and the cytoplasmic fumarate hydratase sits in the cytoplasm, and what it does, it actually participates in the urea cycle, and it does the same as what it would do in the mitochondria, but it does it outside of the mitochondria. It does it in the cytosol, and it uses fumarate to hydrate it into malic acid and actually keep the urea cycle functioning in the cytosol. And this cytosolic reaction is just identical as far as the catalysis and the catalytic steps go, yet it's an irreversible process. So, and it's because of the free energy change between the other participants of the urea cycle. So it seems that fumarate hydratase is very important in the TCA cycle to actually keep the Krebs-Senioji cycle turning over, but it's also very important in urea cycle, where actually amino groups and urea is produced, and eventually uric acid is used to dispose amino groups through the urine. And this cytoplasmic reaction and the balance of the cytoplasmic as well as the mitochondrial reaction is where I will lead the topic eventually. But what I'd like you to take home is the message that there is a cytosolic fumarate hydratase, and there is one in the mitochondria. And those two reactions are very critical as far as maintaining proton or hydrogen balance in the cells. Now, the difference between the two fumarate hydratases as far as the source of water is that in the cytoplasm, fumarate hydratase uses drinking water or interstitial water or plasma, water that we drink, because the cell transports water from outside, from the extracellular space, and the cytosolic fumarate hydratase will use water that comes from the water pipe. Why? And interestingly, if there's a problem with the cytosolic fumarate hydratase enzyme, or the reaction is not taking place at a sufficient rate, actually there are cysts that develop. Mostly we see these in the kidneys, and there are deficiencies in the urea cycle as what we would expect. But interestingly, only cysts are formed and never cancer. On the other hand, if you look at the mitochondrial fumarate hydratase, which uses this metabolic water, which is not from the drinking water, what you see is that the water source is the process that is taking place through cytochrome C and complex 4, where the need of oxygen to produce water is evident. And this metabolic water is recycled in the matrix to produce malic acid and keep the cycle over, but this water is metabolic water. And interestingly, if there's an insufficiency in this metabolic water production, or there's a mitochondrial fumarate hydratase mutation, the result is cancer. And actually this cancer is very aggressive. And I'm going to talk about this biological phenotype a little bit in more details. But what I would like you to emphasize, what I'd like to highlight is that the water source of the cytoplasmic fumarate hydratase reaction and the mitochondrial fumarate hydratase reaction is very different. One is the mitochondrial water or matrix water, and the cytoplasmic fumarate hydratase uses free water or water that we drink. So the question what a biochemist would ask is, why mitochondrial fumarate hydratase insufficiency causes cancer, especially in the kidneys? And is there any way we can answer this question in a sufficient way that we may have some predictions of what is the role of this water or metabolic water production and or recycling through NADPH and how it's involved in cancer development? I think as far as the post-genomics or metabolomics or biochemistry or functional biochemistry-driven research era is coming, these questions are more and more important. Because actually, if the mitochondrial fumarate hydratase enzyme is mutated, the result is cancer. And these are actually kidney cancers injected into nude mice which actually have a fumarate hydratase gene mutated, which means that these tumor cells cannot hydrate fumarate, and there is about a 70% decrease in fumarate hydratase activity, and sure enough, a very aggressive cancer develops, which is what we call a clear cell kidney cancer, which I'm going to talk about in more details, because this is a very important phenotype. And we got this cell line or this model from the National Cancer Institute from Bethesda, Maryland. And this happened is just in 2009. I'm sitting in the lab and I get a phone call from the National Cancer Institute from Dr. Linehan, who runs the surgery, the kidney surgery or urology surgery program in Bethesda. And he told me that they need stable glucose isotope studies because they don't really know what this cancer phenotype is, because a mitochondrial enzyme mutation should not cause cancer. They just had no idea why a fumarate hydratase mutation in the mitochondria would actually produce a very aggressive cancer phenotype. So we agreed to look at this disease, which is a fumarate hydratase point mutation in the mitochondrial fumarate hydratase gene, which is on chromosome 1. The disease is called the Reed syndrome. It's called familial leiomyomatosis, and it appears in the skin and also in the urinary bladder. And I have to tell you, this disease is just fascinating from any aspect that I'm more and more finding out about it, is because actually the leiomyomatosis in the skin is nothing else but muscle cells which actually move our hair follicles to actually preserve heat in our skin. Those smooth muscles actually become or signal as hypoxic or not able to produce ATP, obviously because of the mitochondrial fumarate hydratase mutation. So these patients have goosebumps all the time, which is truly fascinating. And even it's so inconvenient and so painful that some commit suicide, which is really a very interesting clinical consequence of the disease itself, or the fumarate hydratase mutation. It's a rare autosomal dominant disease, so you can inherit it. It has these leiomyomas, and after 10, 15 years of leiomyomas, there are kidney cancers that develop. There's a point mutation in the fumarate hydratase gene in the mitochondria, and there's a more than 70% decrease in fumarate hydratase catalysis rates. And those affect the mitochondria primarily. And there's fumarate accumulation in the cytoplasm. So when we look at these cells, obviously when fumarate cannot be used through fumaric acid cannot be used through fumarate hydratase, there's a fumarate accumulation in the cytoplasm and also in the mitochondria. And many believe that those are actually involved in tumor development. The tumor itself is a clear cell renal cell carcinoma, and I'll tell you why they call it clear cell. Excuse me, it's a cortical tumor, and it's a malignant epithelial cell-based tumor with a clear cytoplasm, and this is how it looks like. It's really a very unusual cancer type simply because you see these very large cells filled with this very clear material. You see many morphological changes in the nuclei that are characteristic of cancer. But if you look at the normal kidney and you compare it with the tumors, what you see first is that there are these huge clear cells in the kidneys, and this is why it's called clear cell kidney, clear cell renal cell carcinoma. If the fumarate hydratase mutation is the background of this kidney cancer or kidney tumor, it's very aggressive, meaning that the patients don't really live more or longer than 6 months if their cancer takes off. And when they operate, by the time they operate, usually these patients have metastasis in the liver and the lungs. So once fumarate hydratase is mutated, fumarate accumulates and fumaric acid accumulates, and there's very little malic acid in these cells. In fact, also succinic acid accumulates, and also every product or precursors that lead to malate formation accumulate in the TCA cycle or in the mitochondria, while malate concentrations decrease. What's also interesting, and this is how they explain the epigenetic events that are associated with this fumarate accumulation, that is, fumarate accumulating in the mitochondria can actually leave the mitochondria through various transport systems and actually can inhibit cytoplasmic enzymes. It can actually activate this hypoxia-induced factor 1 alpha, which is involved in this leiomyoma development, which actually causes these goosebumps or heat reaction or cold reaction in the skin in these patients constantly. And this is a hypoxia. These cells all signal constantly like there is hypoxia, but there's actually sufficient oxygen. The only problem is they can't really recycle the product, which is water, of oxygen and hydrogen in the TCA cycle. So the water use is not sufficient, and they can't really turn the cycle over. ATP is low. They constantly signal as a hypoxia factor, but it's really not hypoxia. It's actually a fumarate hydratase mutation, so they can't really turn the cycle over. So low ATP signals that there's hypoxia, there's not enough oxygen for water production, yet there is water. In this case, fumarate hydratase is mutated. It's a non-specific succination of cysteine residue of proteins. This is also claimed in the molecular mechanism of cancer development. And this was published sometime back in PLOS1 in 2013, August, with this group. I'm also part of this study. And there's elevated levels of superoxide and peroxide formation in defective mitochondria, which are various mechanisms involved in the epigenetic events. Now, myself and my colleagues have been stunned when I talked to Dr. Linehan from the surgical urology service from the National Cancer Institute. when he told me that, Laszlo, the thing is, once we restore this fumarate hydratase, the tumor is gone. I'm like, no kidding. How? What do you mean by that? And this is, just take it literally. If once we restore fumarate hydratase in these tumor cells, that means once we insert the normal gene in these tumor cells, previously tumor cells, they call the tumor cell UOK262. It was from a kidney cancer patient. One was from a primary tumor and the other one was from metastasis of the same tumor. But once they inserted back the normal fumarate hydratase gene, the tumor was gone, meaning that there was no tumor formation and the mice were happy. And indeed, once they fixed this one single mitochondrial enzyme in the TCA cycle, mice were just as happy as ever, meaning that there was no tumor formation. In fact, they couldn't even palpate the tumors. The tumors were not even, the tumor cells, previously tumorigenic cells, were just not able to form tumors anymore. So they published this in Cancer Cell, which is a Nature type publication, very influential. And this was before I got involved in these studies. As you can see, the tumor cells which have the fumarate hydratase mutation in the mitochondria called UOK262 have fumarate or fumaric acid concentrations in the mitochondria about 80-fold higher or more than fumarate in UOK262 cells, which are Y-type, meaning that the enzyme was fixed. That's not enough. Once they actually injected the UOK262 cells into the nude mice, you can see the growth rate or the tumor volume after looking at 30 days, 60 days, 90 days, and after 3 to 4 months, these animals were actually unable to live any longer because of the tumor burden. But once they fixed the fumarate hydratase gene, what you can see here is that there was no tumor formation, and the tumor, then these animals were living just a full, happy life. And more interestingly, when they looked at the mitochondria in these cells, this is a PET scan from an individual with the cancer, and these are very early liver metastases. But what was even more interesting is they looked at these tumors using electron microscope, and what they were discovering is that if there is a fumarate hydratase mutation in these UOK262 cells, you can see how large these mitochondria get. They actually go through what we call a hydropic degeneration, meaning that water accumulates in these mitochondria. Obviously, because of the lack of metabolic water used in the mitochondria. Yet, if they actually inserted the gene back to the the normal mitochondrial fumarate hydratase gene, you can see how normal looking mitochondria are. And I made a bigger magnification just so you can see. This is from their paper. You can see how normal mitochondria is when the wild-type fumarate hydratase gene is present in the cells. And in fact, besides using a fumarate hydratase mutated cell line, and once they inhibited actually chemically succinate formation, which is the precursor of fumaric acid, you can see that mitochondria was again blown up. So obviously the succinate fumarate axis or succinate fumarate transfer of protons in the TCA cycle and the metabolic water use is a key event in these tumor cells to actually restore the normal metabolic phenotype, where actually mitochondria look normal. And you can see these mitochondria still have the cryptase, so there are actually crypts in the mitochondria which actually are pushed because of the water pressure increase in the mitochondria to the outer membrane. There are still some cristae that are maintained, so this is matrix water. So this is mitochondrial matrix water that is accumulating in the mitochondria. So the question is, again, by a biochemist that after seeing all these interesting findings in these tumor cells, what is the relationship between cytoplasmic and mitochondrial fumarate hydratase and their water use as a target to control cancer development? Because as I told you, when actually fumarate hydratase is mutated in the cytoplasma, there's no tumor. There is cyst formation because water accumulates, but there's no tumor formation. So the question is there any way that we can understand what is the main difference between the two enzymes or the fumarate or the water that they use? So one mutation causes cancer, the other one is only able to cause cyst formation in the cytoplasm and in the kidneys. So this is the second part of the story where actually Dr. Szent-Györgyi and his discoveries come to the forefront, and that is practically his discovery of that actually fumarate hydratase in the mitochondria adds matrix water to fumarate, and by then it actually uses metabolic water to actually hydrate fumarate and produce malic acid, which is, he didn't know at that time there's a TC cycle, but his discovery was that water is produced from food using this fumarate hydratase reaction, and the hydrogen comes from food and the oxygen comes from the air and through transport through the oxygen carrying system, hemoglobin, and that has to be taken to the cell, and this is how water is produced in the mitochondria. We have to go back to the 1940s when a British scientist called Dr. Mellamy has published a paper where he described what metabolic water is. And he wrote the utilization by the body of ingested food substances and of tissues reserve yields, among other things, quantities of metabolic water. What's interesting is that as the complete combustion of 100 grams of fat produces about 110 grams of water, this is obviously what the hydrogen saturation does, of excess hydrogen saturation in fatty acids, metabolic water, whereas if you use carbohydrates, you only produce 55 grams because the hydrogen saturation is lower in carbohydrates. So, and he wrote that fatty foods are believed to be particularly valuable as protection against desiccation. And this contention would appear to be supported by the fact that many animals which exist in deserts have large reserves of fat. You've seen camels that kind of wander in the desert. Those bumps on the back, that's fat. Those are fat patch or fat pools. And this is what they use to oxidize and produce their own water where there is no water. All animals in the desert, they have to produce metabolic. Well, you have a question? No? All animals in the desert have to produce metabolic water to survive. And obviously this is key to their survival. There's no water. Actually, they cannot even drink water. For example, if you look at the rattlesnake, for example, if you look at the scorpions, if you look at any kind of desert reptiles, they all have to produce their own water. In fact, migrating birds have to produce their own water. That's why they have to fill up with fat before they start migrating. And this is why I brought my Arizona Diamondback boots, because these guys, you can look at it after the talk, but these are Diamondback rattlesnakes. And I had to wait 4 months because what they do, the Indians actually catch this snake. We don't kill the snakes for the boots. They catch the snakes, they throw them in a pit, they feed them, they shed, and then you have your boots. But these guys, they actually have to eat their prey and burn their fat to produce their own water. And because of that, because they don't really recycle the water, they actually use the water for its water role. They cannot really produce ATP, so they have to hibernate. So everything kind of, when it comes to mitochondrial function, everything kind of just explains how phenotype and adaptation actually depends on mitochondrial metabolism, and how the production of water and how the recycling of water through fumarate hydratase is a key event in, for example, cancer prevention, because actually those animals never develop cancer. If you have heard of these marine animals or the reptiles, actually it's very uncommon for them to develop cancer. Even though they have cancer, it's really biologically is not any malignant as far as disease process goes, because the cancer cannot really grow to a large size of tumor, or they cannot really metastasize very rapidly. So actually if you look at fish, for example, if they have you guys been to a Japanese restaurant where they have this fish in the tank with these little kind of colorful little bumps in their heads? That's a KRAS mutation. That's a carcinogenic KRAS mutation. And those fish actually live forever. I think the oldest fish was like 12 years old, and Bruce Lee had one. The Kung Fu guy, the movie actor, he actually had one of those fish. And those are actually oncogenic KRAS mutations. But in fish, because they actually produce their own water and they don't really use the water from salt water, obviously you can't really drink salt water. All the mammal, all the sea mammals, all the fish, all the reptiles, all the migrating birds, they have to use metabolic water to support their water needs. And in fact, when it comes to cancer, the first species that actually develops cancer is the birds. The birds have leukemia. Actually, the first bird's leukemia model was discovered by 1911 by a French scientist called Roux. He got the Nobel Prize in 1963. He was some 80-some years old, because nobody actually believed him, but he could actually transfer cancer from one bird to another using viruses. So and those were leukemias. So what we believe is that this metabolic water use is very key to understand. I'm going to talk about this a little bit more details, but metabolic phenotype is very important and critical to understand how metabolic water use or recycling is involved in the control of DNA, involved in controlling DNA stability. And this is where we understand some species or phenotype-related relationships between migrating birds, cancer, and metabolic water production versus drinking water. Because the advantage of moving to a temperate climate from the desert is that you can actually drink water. You don't have to produce your own water. You don't have to hibernate anymore, because you can actually drink water. But there's a catch to it, which I'm going to talk about in a little bit more details. And for that, we need to understand what's the difference between metabolic water and the tap water, because it's different. So the water that you drink there, or I'm drinking here, I'm taking a sip, is different what you produce in your mitochondria. And simply because in the mitochondria you produce water from fat. And as you can see, the hydrogen in fat has very little deuterium. So when you look at the tap water, which we measure as Vienna standards of mean oceanic water, it's actually 155 parts per million. I'm going to tell you what this number means, which means that the water that we drink has about 150 deuterium labeled water in the entire water pool. And if you look at flour, which is a sugar or carbohydrate, you can see it's close to 155 ppm. Table sugar is 146. But if you go to cottage cheese, and actually when you look at olive oil or even look at butter, what you can see is actually the deuterium content of the hydrogen that is attached to the carbons in fatty acids is just dropping. What's really interesting is if you look at pork fat or cow fat, it's actually 118 ppm, almost a 20% change or close to 20% change. What's more interesting is that if you look at, so the natural fat with low deuterium enters the cycle, and this water that is produced from through cytochrome C, the water that is produced from the hydrogen of fat is lower in deuterium, simply because the fat source that we eat is lower in deuterium, which is actually the hydrogen that is attached to the fatty acid carbons, especially in saturated fatty acids or fully saturated fatty acids. So this water is different than what we drink, and it has lower deuterium that is attached to fumaric acid. So the malic acid that is carrying this water or this hydrogen from metabolic water is lower in deuterium. So the NADPH, or the reducing equivalent that is produced from malic acid that is produced in the TCA cycle, has low deuterium. So eventually, if you use metabolic water for DNA synthesis, your DNA and the hydrogen bonds will have less deuterium, and that has a major physiological consequence that I'm going to talk about in a little bit and share some data with you. But and if that's not enough, if you look at unsaturated fat, deuterium drops further, it's 110 ppm. And if you look at unsaturated plant source of fat at double bonds, actually at high altitudes with low photon pressures, exposed grass, it's only 102 ppm. This is a model. And actually this is probably the lowest that nature gets as far as deuterium load in fatty acids. Now, this is from a French group who actually measures deuterium content of various fat products from plants and also from animals. So this is data from the literature. And one would ask, like, now how would be this possible? Why the fat has lower deuterium than the water that we drink in the form of water? Anyone? Any guess? No? I'll tell you why. It's because of photosynthesis. When it comes to photosynthesis, what happens is when the photon comes and hits the chloroplast, which is actually the organ that splits water for oxygen release and use the hydrogen from water to actually synthesize various substrates and various products in the plant cells, based on the energy of the photon or the light, not all water molecules will break eventually at the same rate. Because if the water contains a deuterium, that bond is harder to break. So eventually there is a discrimination against water, especially in plants that are actually growing in the bottom, or plants that actually have no sun exposure, for example, grass. They actually have less efficiency as far as light comes in. They have less efficiency in splitting water if the water has deuterium. And as the result of that, the NADPH, as the source of reducing equivalent for biosynthesis in plants, will drop deuterium. So when it comes to plants that actually have photosynthesis in the shades or have less light, for example, green, like grass, they actually discriminate against deuterium and water that has deuterium, and eventually other metabolites, other substrate and products are deuterium depleted. Interestingly, this is not true with fruits, because fruits are actually ripening when the sun is the strongest, as summertime, and the light is strong enough to break all water molecules regardless of deuterium or oxygen isotopes in the water. So, and this is why the flour, the table sugar has no difference as far as deuterium enrichment goes, because actually those are exposed to stronger light, and the hydrogen discrimination or deuterium discrimination is not as efficient in the middle of summer when the grapes or the fruits are ripening. So when you look at the animal fat, for example, cow and pig, which eat the grass and cannot really reach the trees for fruits, they have low deuterium in their fat. While other animals, just like fruit eaters, they actually have same deuterium content as what you would expect in the water and also in the fruits. So eventually this biological process of deuterium discrimination will end up in your DNA in response to fumarate hydratase action and metabolic water use. So, as far as using a cellular model of mitochondrial versus alternative NADPH or NADPD or deuterium-loaded NADP production, we have to now separate mitochondria versus cytoplasm. And this is where two major tumor phenotypes appear. One, as I was telling you, fumarate hydratase was one key enzyme which causes kidney cancer, aggressive kidney cancer. Do you guys, anyone knows another? I know you are biochemistry students, but there's this other cancer which is called the glioblastoma. It's a very aggressive brain cancer type, and actually it has a mutation in the isocitrate dehydrogenase enzyme, and it's an NADP dependent. So we know several mutations, enzyme mutations that are related to mitochondrial metabolism which cause cancer, actually determine cancer's phenotype as far as how aggressive they are. But just to go back to a normal cell, obviously, as long as you oxidize fatty acids and you use your mitochondria efficiently, you're going to use or produce a reducing equivalent through mitochondrial metabolism, which has low deuterium. Alternatively, if you use glucose or free water, which has high deuterium concentration through glycolysis, eventually you could produce NADPD, which is deuterium loaded, and that will end up in DNA and the nuclear membranes eventually, but nuclear membranes and fatty acids. But as long as mitochondria is active, low deuterium fatty acid products, or NADPH, will actually, with low deuterium load, will actually be responsible for DNA synthesis. So in your DNA, the hydrogen bonds are fine. There's no deuterium, so there's no difference as far as hydrogen deuterium or deuterium binding, no increase as far as your mitochondria is working appropriately. So what is wrong with deuterium? Why deuterium is so bad as far as its clinical behavior or as far as its physical behavior goes? It's because it's twice as heavy as hydrogen by weight or mass. It's chemically identical, so it participates in every chemical reaction. Deuterium participates in reductive synthesis just like hydrogen would. It changes DNA methylation kinetics because of its different physical properties, and it eventually alters gene expression and likely induces aneuploidy. We know this because cells that can only use, for example, sugar for oxidizing for NADPH generation, these are red blood cells or neurocytes. Those actually can only use glucose and free water from the cytosol. Red blood cells, they have to lose their nuclei to be safe as far as operating in our body. And neurocytes or nerve cells, they have to lose their DNA replication system, so they can't really divide anymore. Their nuclei look huge. They are almost like aneuploid, but they actually have to lose. We've never seen a neurocyte dividing on its own simply because they have to lose this capability. They are locked in a post-mitotic state. And then they can use glucose and they can load their DNA with deuterium, but they have to take some safety measures. Very interesting is that when you look at your bowels or your intestines, your small intestines, there's never tumors. from your colon or from large intestines. It's one of the most common tumor sites. Difference is epithelial cells of your small intestine, they actually can't enter, the water that you absorb can't enter the epithelial cells. They actually have a water pump system which actually sidesteps the cell. So practically they don't even use cytoplasmic fumarate hydratase. And in fact, their fatty acid oxidation is so intense, when you use fatty acids as tracers and you trace them through the gut, you're going to see modified fatty acids all over the place in your circulation, simply because your epithelial cells in your small intestines have a very different metabolic signature or fingerprints that you would expect from cells that have actually capabilities of developing cancer. So we starting understanding this process very clearly now. And this is for the benefit of the clinicians who treat cancer, hopefully in a very short future. So Dr. Sandhyoti proposed that hydrogen carrying the succinate and fumarate, the only role of succinate and fumarate is to carry hydrogen from food. And this was back in the 1920s. And this is actually a cytochrome C and the complex 4 bound process, where actually oxygen is used from air and hydrogen is used from NADH or from the intermembrane space. And this is this ATP synthase, which I was going to tell you a little bit more about. The hydrogen that is pumped through complex 1, complex 3, the majority of hydrogens is pumped through complex 1 and complex 3. They actually return through ATP synthase because there's a gradient. And when the hydrogen returns, then ADP and ATP conversion occurs. So ATP is synthesized, and this is why we don't have to hibernate. This is why we can go on a ski trip in the middle of the winter, because we can just use this process to generate our own heat in the form of ATP. You remember reptiles, they remove this water, they don't really recycle it, and therefore they have to hibernate because they can't really run this ATP synthase as efficiently, simply because they have less oxaloacetate to keep the cycle turning over and producing oxidizing fatty acids. They use this for water, the fatty acid hydrogen from water, instead of recycling water into their metabolism, TCA cycle. Now, Dr. Sandhyoti, in a television interview in 1973, and I was actually watching that television interview. You probably haven't because you're too young, but I was actually I was 11 years old, and I was fascinated. I mean, this guy came back home from America, from the United States. He was chased out by the communists from Hungary. He came back from the US and he gave this television interview. And he was talking about what he thought about cancer. And he said, actually, it's probably not these big clumsy proteins, which are not really reactive, but it's probably electrons that play a major role in cancer development. And I was fascinated. I mean, he was talking to this reporter, and he was talking and giving this. I was always very interested in science, especially after my grandma was telling me all these stories about Dr. Sandhyoti. But this story was, to me, just fascinating. Now, I was not the only one who was watching this TV, because a biologist named Dr. Gabor Somlyai, back in 1976, and I don't really know how old he was, I think he was just getting into college, he wrote a note for himself back in 1976, 3 years after this television interview, where actually he said, it's a handwriting, it's Hungarian, so we actually translated it and wrote it here. So he wrote that Albert Sandhyoti tried to explain cancer at a submolecular level. He assigned a major role to the electron. I think, and this is his writing, hydrogen ions have, besides numerous other factors, the decisive role in the development of cancer. Hydrogen ions consist of one proton. The diameter of an atom is about 10 to the minus 8 centimeter. That of the nucleus is 10 to the minus 12 centimeters. So in case of the hydrogen ion, positive charge is distributed on a much smaller face, which is what we know is the Columbian effect. If in a cell hydrogen ions get out of balance for any reason, proteins or other substances can be deformed to an extent which leads to uncontrolled cell proliferation. And this was his note. He put it in his pocket or in his drawer, and that was it. So when I got to the United States, I joined the UCLA faculty in 1998, and just a year before that, I published a paper in Medical Hypothesis. In the same year it came out, but we started writing the paper in 1995 with my colleague. It was published in Nature, Drug Discovery, and what the author, who is at MIT, my colleague, what he mentioned about this paper was that this study was among the first modern studies to track carbons in cancer cells and called into question common assumptions about cancer metabolism. And this paper deals with the non-oxidative pentose phosphate pathways and glucose 6-phosphate dehydrogenase. And what's really interesting is that when we were studying anti-cancer drugs for Novartis in this particular case, I published a paper in the New England Journal of Medicine in 2002, where actually I described NADP or NADPH as one of the major molecules that actually Gleevec controls. Gleevec is a very efficient targeted therapy in cancer. In fact, in chronic myeloid leukemia, is almost curative. So as long as patients get this Gleevec, which is a tyrosine kinase inhibitor, the authors in the original paper of this New England Journal of Medicine described all the molecular mechanisms, and I added the metabolic mechanisms. And in fact, NADPH, as we named it, in the oxidative branch, produced in the oxidative branch of the pentose cycle, is what played a key role in disease control using this very efficient targeted anti-cancer drug. So it seems, after all, that it doesn't matter how you look at it, if it's cancer epidemics, if it's actually an efficient cancer treatment, if it's a thought of a medical student, or actually coming from a Nobel Prize winner, Dr. Szent-Györgyi, it seems that metabolism really is key to understand, especially hydrogen and its deuterium part is key to understand how the process goes in tumor cells and how to control tumor cell metabolism, yet how to control tumor cell phenotype. And just to show you what Gleevec does is that actually it increases NADPH production in the TCA cycle. In the meantime, it controls the oxidative branch of the pentose cycle, which also produces NADPH, but from tap water. So it switches water use from tap water or from drinking water to mitochondrial matrix water, and hence there is a control of tumor cell proliferation, a complete control of tumor cell proliferation, so much so that these patients don't qualify as leukemia patients anymore. It's a very effective treatment in BCR-ABL positive or Philadelphia chromosome positive leukemias. And you're going to hear about this when you're studying hematology. But anyways, there are a number of reactions that produce NADPH. Some, and almost all of them, produce NADPH using tap water or glucose or sugar, which you can see here. And there's also mitochondrial NADPH production, which is deuterium depleted. And this is the difference between these two NADPH molecules. This carries less deuterium into DNA, and this carries more deuterium into DNA. And we have discovered a very recent cycle called the serine synthesis and glycine oxidation. We published this in Nature. It also uses NADPH. When it synthesizes NADPH, it also uses drinking water. So it's really, mitochondria is the only cell organelle that can actually reduce or decrease deuterium in NADPH. So this is our paper from the Journal of Biological Chemistry from 2001, and this is the Gleevec, or the effective leukemia treatment with increasing doses, and sure enough, these cells start oxidizing fatty acids. So the metabolic fingerprint of a successful cancer treatment is oxidizing more fatty acids in the expense of sugars. So if you remember, I told you fatty acids have very low deuterium. So in these cells, metabolic water is produced more efficiently, and metabolic water with low deuterium is recycled into NADP through NADPH into nucleic acid, DNA, and nuclear membrane structures. Now, if you looked at enzyme activities, sure enough, glucose 6-phosphate dehydrogenase, which is one of the NADPH deuterium loading enzymes, is actually decreased dose dependently and significantly. And so is fatty acid synthesis, or K562 cells, which are leukemia cells, show a decrease in DNA synthesis, in DNA aneuploidy, in nuclear membrane synthesis production, and they start oxidizing fatty acids in their mitochondria. So it seems that actually it's a number of metabolic events in response to this tyrosine kinase inhibitor, which eventually accomplishes one very important task, and that is converting or reversing the tumor cell phenotype back into a normal myeloid leukemia, normal myeloid blood cell. So these leukemia cells are not able to proliferate any longer, and this is what we published in the New England Journal of Medicine. So what happens if we treat tumor cells with water which has less deuterium? This is what happens. If you look at MCF7 tumor cells, which are breast cancer cells, A549 cells, which are lung tumor cells, and HD199, I believe these are colon tumor cells. If you actually treat them with less deuterium, you can see that's how slower they grow in culture. Why is deuterium so different than hydrogen is when it comes to growth conditions? Because deuterium has one extra neutron. It has a mass of 2 daltons instead of the 1 dalton, which is hydrogen. And we know that it has a proton, an electron, and a neutron in the case of deuterium. And these are the water molecules that you drink in your water. You can have H2O, HDO, and you can have D2O. So oxygen that has 2 deuteriums. As long as the NADPH source is your drinking, the oxidation branch of the pentyl cycle, or the SOGC, the serine oxidation glycine cleavage system, then you use either this hydrogen or deuterium to produce NADPH. While if it's the mitochondria, then your hydrogen doesn't come from water, it comes from fatty acids through cytochrome C. Now, if you look at the water molecule that has a hydrogen bond to oxygen, that has a vibrational frequency of the bond itself, which is about 3400 value. And then if you look at deuterium, actually the binding is shorter and the resonance is different. The isotopic rate changes are related to this vibrational frequencies, and this is why this bond is so hard to break via sunlight, and this is how photosynthesis discriminates against deuterium. Now, if you look at the hydrogen and carbon, hydrogen nitrogen, hydrogen oxygen binding, you can see that there is, to break this energy, once it's replaced with deuterium, it actually gets slower. If it's deuterium instead of hydrogen, actually if it's a carbon and deuterium binding, it's 7 times slower than if it was hydrogen. If it's a nitrogen deuterium binding, it's actually 8.5 times slower, the kinetics of the reaction. And if it's actually oxygen and deuterium, it's actually more than 10 times slower than if it was hydrogen. Now, if you actually change carbon to a stable isotope of carbon in the binding, it only changes this binding kinetics by about decreases it by about 1.4 times slower. So actually the largest isotope effect occurs when deuterium is introduced into a biomolecule. And this is actually very significant of a change. So this is what happens. When an enzyme comes or the photon comes, it really cannot really lift deuterium that easily. It cannot really break that bond very easily. So actually deuterium loading into metabolism is a ticking time bomb. In fact, if you look at the plasma, and this is very, I would say, unknown to even clinicians, is actually deuterium in your plasma or your blood is the highest as far as its concentration goes. It's actually 12 millimole per liter. If you compare this with calcium, magnesium, potassium, actually deuterium is the highest of all, including glucose. So deuterium is really a very common and it's a very important anorganic ion that actually regulates cell metabolism. For example, if you look at sodium and hydrogen exchange, and this was published in the Proceedings of the National Cancer Institute, the National Academy of Sciences, we call it PNAS, back in 1984, what they observed that actually hydrogen transport from the cell has a preference to pumping hydrogen in the expense of deuterium, and as deuterium accumulates in the cells and hydrogen is getting pumped out, actually there are cell divisions, but that's not enough. If you look at hydrogen pump or fibroblasts that are actually transfected with this ATPase gene from yeast that actually pumps hydrogen, and there is no sodium exchange, so it's a more efficient hydrogen transporter. What you can see is there is actually tumor growth. So you can actually transform cells by changing the hydrogen deuterium balance in the cell. Some believe it's a pH-related matter, some believe it's actually a growth signal. But a paper back in Nature in 1988 was reporting that if they actually transfected fibroblasts with a yeast ATPase-dependent hydrogen transporter, those cells became cancerous. So what they were saying that actually in the conclusion was that the yeast ATPase gene behaves as an oncogene in mammalian cells. These experiments support a crucial role of intracellular pH in the growth control of animal cells. We know it's not only pH now, we know it's actually deuterium loading into DNA. So we have to come up with a different model, and this is actually patients drinking deuterium depleted water. If you actually deplete water in deuterium in water, you can decrease free water deuterium in your cells, and so you can actually reverse some epigenetic events and aneuploidy, decreasing deuterium. Deuterium actually gets in from water through the oxidative branch of the pentose cycle, and for that we proposed a new model, and we said, we know in mitochondria, in tumor cells, mitochondria is not working. This is the first critical metabolic event in tumor cells. Mitochondria is defective. We know there is no oxygen, tumor cell hypoxia. Doesn't matter what the cause is, eventually mitochondrial metabolism decreases. So deuterium depletion decreases for the cell. And the reason I showed you the fumarate hydratase model, because that's the most dramatic decrease in fumarate hydratase activity. But as long as hypoxia goes, as long as substrate deprivation goes, actually it is the same as far as decreasing mitochondrial deuterium depletion. So once the deuterium depletion process in the mitochondria is inhibited, then free water and glucose-based high deuterium labeled substrates start loading DNA with deuterium. And as a result of that, the high ppm deuterium or high concentration of deuterium from food and from free water gets into the DNA, changing all these binding kinetics and changing the protein as well as the nuclear membrane characteristics. And the result of that is aneuploidy and eventually cancer development. And this is exactly what happens in these tumor cells, which we studied with the National Cancer Institute. And don't look at the figure yet because it's a little complicated. I'll tell you what happens here. So this is the wild-type fumarate hydratase cell line. You remember the one where we actually reinserted the normal fumarate hydratase gene. This is the one. This is actually the normalized tumor cells. So we set it up as 100% for this study. We had an empty vector because you always have to do your control, which does not have the gene, the fumarate hydratase gene, but it has the vector. And this is a primary tumor, and this is a metastatic tumor, and this is a primary tumor from the same patients. These have the mutated fumarate hydratase. Now you can see here there's glucose consumption is increasing, and this is what you would expect from a tumor cell. Some of it is almost like almost double as far as glucose consumption. You can see that glucose oxidation is not very efficient. Yes, indeed, because mitochondrial substrate oxidation is going down. And this is a 13C tracer glucose study, which I'm probably not gonna have much time, but we're gonna mention. What's really interesting is that these cells are producing lactate. This is the Warburg phenotype. Tumor cells that actually use glucose and they cannot oxidize pyruvate, they're actually going to produce lactate. This was described by Dr. Warburg back in the 1950s, and ever since we call it the Warburg effect, because it seems that tumor cells use glucose and they produce lactate for a very sluggish ATP gate. You can also see that glutamine does not get labeled, but what you can see here is that fatty acid lignoserate is accumulating in these tumor cells. And I'm going to talk about what lignoserate is. It's a C24 saturated fatty acid. And so is nucleic acid labeling RNA concentration, which you would expect from tumor cells. So obviously we have a tumor cell model where fumarate hydratase is not working, and there's aneuploidy, and what happens if we actually decrease deuterium in water? So we actually trick these tumor cells and we tell them, listen guys, we don't care if you use your mitochondria or not, you're gonna get low deuterium water anyways. So you can't really load your deuterium, your DNA and RNA with deuterium. And this is what happened. We're gonna present this in San Diego in about 2 weeks. So this is why I'm not going to talk much about this study. I'm just going to, because we are under an embargo right now, but I can show you just a little bit of data, which is lignoserate, which is the same fatty acid as accumulates in kidney tumor cells. Deuterium depletion in water in another tumor cell, like Mia-Paca cells, actually decreases dose dependently when deuterium is depleted in water. So is nuclear cholesterol. Cholesterol in MCF7 cells, which are tumor cells, cholesterol is just as important as a saturated fat product or sterol ring to actually make these fatty acid membranes very rigid to hold these lot of DNA in these tumor cells, which we call aneuploidy. So deuterium depletion in Mia-Paca cells decreases the same fatty acid, lignoceric acid, from the nuclear membrane, which is a long-chain saturated fatty acid, to prevent aneuploidy, and the same that happens with MCF7 cells as far as cholesterol synthesis goes. I can't show you more because we're going to present this in San Diego in about 2 weeks, and this is all I can show you for now. Now, what is lignoceric acid? It's a saturated long-chain fatty acid which actually forms 30% of nuclear membrane. It actually makes the membrane rigid and able to hold a lot of DNA together. It's more like a tennis ball compared to a soccer ball. It's actually it makes the nuclear membrane very stiff, and it helps to maintain aneuploidy in tumor cells. So actually in some tumor cells, this can increase up to 60% to make these membranes very rigid. And it's the same with sterile saturated fatty acids linked with cancer. This is what we believe is probably the most key element of the story. This is fatty acid synthase, and you're gonna learn this for your exam. It's a huge enzyme complex. It synthesizes fatty acids from acetyl coenzyme A, and I'm gonna show you how it works. But let's go look at the enzyme itself. So what's really funny about this enzyme, it synthesizes palmitic acid, which is palmitate. It's a 16 chain length fatty acid, saturated fatty acid, which actually leaves this chamber after 16 carbons, and it gets to another enzyme complex called fatty acid chain elongase. However, the way how the reaction goes is actually anchoring the ACR carrier protein and actually adding acetates to the growing fatty acid chain. And obviously, this is a mammalian fatty acid synthase, and this is a bacterial or fungal fatty acid synthase. And what's really just a very important difference between the two fatty acid synthase systems, that this guy is locked in as far as the size of its reaction chamber, so it can really synthesize only one kind of fatty acid, palmitate. This guy is a little bit different, and this is because the reaction chamber is open. And this is from the Scripps Institute from San Diego, La Jolla, so they are not very far from us. But anyways, the mammalian cell, or the cells that actually can develop cancer, have this kind of fatty acid synthase. While actually fungal or bacterial cells, they grow continuously, and obviously their deuterium concentration would not change the rate of fatty acid synthesis whatsoever. Once you withdraw them from the cycle or give them no nutrients, they actually form spores. So this fatty acid synthase, as far as its flexibility or variations in conformation, is not very broad. Yet this guy here, if you look at electromicrograms or I think these are diffraction pictures of the enzyme itself. Here you can see the reaction chambers, and this phenotype is very interesting, this TE2302A, because listen to this. If you actually mutate a protein in the enzyme itself, it opens up this reaction chamber, and what happens, it rapidly elongates acid chains into C16, which is palmitic acid. But that's not enough. It actually cannot release this product, but it will start elongating it into ACR chains, which are actually longer than C16. So, bummer, there is a fatty acid synthase which can actually synthesize longer fatty acids. You just have to open up this reaction chamber, and they can actually fit larger fatty acids or fatty acid chains in the reaction chamber for synthesis and elongation. Sure enough, this unassigned region, yet you know that this region of the fatty acid synthase which can open is actually unassigned. We believe that actually mutations as well as pH and deuterium load can actually modify the size of this reaction chamber. So eventually, the fatty acid synthase, which is associated with mitochondria, with this Golgi apparatus which hangs out and hangs with the mitochondria, we know that this reaction chamber has extreme flexibility. It can actually elongate these long chain saturated fatty acids for membrane formation in tumor cells, and this seems to be one of the sites where DNA as well as pH regulation may take place. So we have in place in tumor cells a number of key components for aneuploidy. DNA load, because of mitochondrial defects, altered fatty acid synthase, which actually does not stop at long chain saturated fatty acids, but it keeps elongating them, forming nuclear membranes which are more rigid, and eventually a continuous cell signal in aneuploid cells, and a very different and altered gene expression. So I'd like you to take home some of the messages that we were talking about here today. And one is impaired mitochondria is involved in cell transformation, and we knew this, that there's nothing new about this. We know mitochondrial metabolism is very key to decreased mitochondrial metabolism is key to tumors development. There are actually deuterium loading, there are actually natural water-dependent heavy DNA-producing metabolic reactions, and these balance have to be very closely regulated for DNA structure as well as for DNA stability. Extramitochondrial NADPH synthesis opens a new window or therapeutic window for deuterium depleted water to maintain and or restore normal cell function. And we knew this, that the oxidase branch of the pentose cycle for a long time, or longer time, we knew those are actually therapeutic targets in cancer. Excuse me, we did not know that it can be affected so dramatically using deuterium depleted water. This is what we're going to present in San Diego. So, don't tell this to anyone. So, our approach provides a mechanism regarding lipid-based ketogenic diets. For example, because this is what happens from a Florida group, they published this paper in PLOS when they say, oh, we feed these animals with fat diet, and actually if we put them in high oxygen pressure or hyperbaric treatment, their tumors are gone. So obviously it's metabolic water, because fat brings the low deuterium and oxygen treatment increases metabolic water production in the mitochondria, and I'm going to talk to them in San Diego because we already got in touch. But anyways, it seems that phenotypes, dietary modifications, and hyperbaric oxygen treatment can actually explain some of the findings, animal models that are now entering the medical literature. So mitochondrial fumarate hydratase seems to be in cancer less efficient than deuterium loading enzymes, especially the ones that are involved in DNA production through cytoplasmic fat fumarate hydratase. So this Peter Tudor is just bending to the mitochondrial side. Yet if we actually ease mitochondrial metabolism with deuterium depleted water, which lifts the deuterium load through both arms, the mitochondria as well as the cytoplasmic fumarate hydratase. Mitochondria doesn't have to be fixed, but cytoplasmic fumarate hydratase gives us the window and the oxidase branch of the pentose cycle to actually lift this elephant off the shoulder of cellular metabolism, and eventually hydrogen takes over and aneuploidy can be controlled and reduced in tumor cells. So the newest biomarker that we are developing for cancer is determining the NADPH-NADPD ratio in tumor specimen, just to see how efficiently mitochondrial versus cytoplasmic deuterium loading mechanisms are balanced. Because this balance is very critical as far as cellular metabolism goes and DNA stability goes. Mitochondrial natural deuterium depletion through fumarate hydratase, and this is the key enzyme in using metabolic water, is fumarate hydratase, and this is the key to natural deuterium depletion, which occurs naturally in the mitochondria versus the one that occurs through cytoplasmic food or deuterium depletion through water. And easing that up, the process up, we can actually influence tumor cell metabolism. And I think we're almost close to finish our time. So I have to thank Professor Dukes for giving us the opportunity that we can spend time together talking about these topics. Dr. Shomyai, who is involved in deuterium research in cancer. Tracy is our technician. Dr. Pauly is my colleague at UCLA, and Dr. Linehan is at the National Cancer Institute, who actually provided the model with the fumarate hydratase mutated phenotype. And with that, I want to thank you for your attention.